So this lecture is going to cover some of the commonly used augurs in microbiology class. So if you'll recall from lecture, we've talked about what a selective versus differential augurs are, what general purpose media is, what a chemically defined media is. So we're going to go ahead and talk about some of the individual types of media that you'll find in microbiology class, as well as how they're used for diagnosing different species and things like that in a hospital setting or in a lab setting. So one of the most common types of media that's found in a microbiology lab is something called nutrient agar or TSA agar. Nutrient agar or TSA agar, nutrient agar is its own thing. This stands for tryptic soy agar. And essentially what this is it's a petri dish. If you recall that our petri dishes have an agar inside of them that contains an agar, kind of acts just like the jello. You can have lemon flavored jello, strawberry flavored jello, jello with chunks in it, anything like that. So the agar itself is just that stuff that comes from the seaweed, the solidifying agent, and whatever you put in the agar, the flavoring of your jello, if you like, the agar is the jello, gives you different types of media. So this is a really, really, really general purpose, a very, very, very generic form of media. This type of nutrient agar or tryptic soy agar, these are both different types of agar, by the way, but they do pretty much the same thing and they work pretty much identically. They look the same. Essentially, all they are is you take a beef broth or chicken broth or vegetable broth, in this case, tryptic soy broth, and they boil a bunch of soybeans or boil a bunch of bones and meat and things like that until they get a nice soup off of it. And then they take the soup and they filter the soup. And they run it through a bunch of filters and things to get all the bacteria out and all the junk out. And then they mix it with the agar, the jello, and this is what you get. So this is essentially beef jello or vegetable jello. It's a very, very, very general purpose media. It has full of proteins. Bacteria love to eat proteins, and there is a ton of proteins inside of this nutrient agar. Pretty much any type of bacteria species that you put on this plate will grow, hence the type it's a general purpose. Gram-positive bacteria will grow, gram-negative bacteria will grow. Pretty much any type of species you put on here will grow. You will not be able to tell anything about these guys from what they are. It is not a differential or a selective media. It is a general purpose, all-purpose media. Pretty much any bacteria species will grow, and unless you know what you're looking at, they are all pretty much going to look the same. There's a lot of yellow bacteria out there and a lot of white bacteria. This is some sort of staph species. This is some sort of micrococcus species. But up until that, you can't really tell what they are. You just kind of have to know what you're looking at or do further tests. So general purpose medias, nutrient agar specifically, is just something that people use a lot in labs just for, I need to grow something real fast, or learning how to do streak lines, or learning how to work with augers and things like that. You're not going to see this one as much in hospitals and things. Not a lot of bacteria are going to love this one that are specifically found in humans. A lot of human organisms are fastidious. If you recall, that means they like specific, or they require, specific growth factors in their diet. And this type of agar, these nutrient agar, these general purpose medias, they sometimes lack that. Streptococcus species don't really like to grow on nutrient agar very much. They will grow on it, they just grow extremely slowly. So what you would do is you would take a bacteria off the sample, off of your patient or whatever. If you were going to use this type of agar, you probably wouldn't for a patient. But you'll just put a little streak on there, put it in the incubator, and let them grow. Now, the common incubation time for microbiology plates is anywhere between 18 and 24 hours. The closer to 18 that you get, the better. And that's a more reliable result. Anything after 24 hours is not legally supposed to be used for diagnostic purposes in a hospital. You have to throw it away and start over. And I'll talk about why in just a second. So tryptic soy agar, nutrient agar, very general purpose, generic media. It's full of proteins that the bacteria like to eat, and that's what they're going to eat in this type of agar. Other types of agars have different things added to their jellos. Different types of medias have different things added to the jello, and that's different things that bacteria will eat. But on this one, bacteria that will be growing on this type of media will be eating the proteins that are found embedded inside of that agar. So nutrient agar, in this case, the nutrients are proteins that are either going to be coming from beef broth, or in the case of tryptic soy broth or tryptic soy agar, vegetables, something like soybeans and things like that. So this is A non-chemically defined agar, tryptic soybeans. I don't know what the chemical definition of a soybean is. Don't know what the chemical definition of beef broth is because it changes from soybean to soybean or probably from a batch of broth to broth. So this is a non-chemically defined media. So the next media that we'll talk about is mannitol salt agar. And now I've got YouTube videos for you guys on here, and this is someone going over how this agar works as well. So now we're going to start getting into our differential and our selective agars here. So this is when things are going to get slightly difficult or complicated, but it's also fairly straightforward. So these types of agars, especially mannitol salt agar, it is selective and differential. So it is selective, which means it kills some particular species of bacteria while allowing others to grow. And it's differential and means that the bacteria that are allowed to grow on this particular plate, you can tell them apart based on some sort of characteristic about what they do. Now, almost all agars on the microbiology world work on the concept of what bacteria eat and what they produce as a waste product. So, on one front, you will be looking at what they're eating, or do they have the ability to eat this particular substance? And if the answer to that is yes, that's one particular species. If the answer to that is no, it's a different species. And how do you know if they have the ability to eat this? Well, you're going to look at what they produce as a waste product. They eat a sugar, they produce X. They eat this, they produce that. And that's what you're going to look for. You're going to look for the products that these organisms produce. Okay, so this first one that we're going to talk about is mannitol salt agar, or MSA. Now, mannitol is a type of sugar. It's used a lot in diabetic candies. It's not the easiest thing for our bodies to digest. We can eat about 5 or 6 of these in about an hour and a half period. 5 or 6 little gummy bear sized things that contain mannitol, but pretty much anything other than that, it's going to turn into sugar alcohol in your stomach. And you're going to have a really bad time afterwards. But anyway, some species of bacteria are able to eat the sugar mannitol, not others. So some of them are and some of them aren't. And that's one of the key characteristics that we'll be looking at here. The other thing that's found in this agar is salt. Lots and lots of salt. 7.5% NaCl, sodium chloride. And you have to put the NaCl part because there's other different types of salt. So you have to specify. So mannitol, the sugar, and 7.5% salt. Now, 7.5% salt is a lot of salt. The salt content of your blood is about 0.9. 0.9, so way low salt content in the environment, and 7.5% salt is a lot of salt. So the bacteria that will be able to survive in the presence of that high salt content will be able to grow. And the bacteria that are not able to survive in this high salt content, their cells will dry out and that little organism will die and desiccate. So that's kind of a concept of how this agar is going to work. So the very first thing that we're going to talk about with this agar is that it is selective and differential. Okay, so this is how it looks like with nothing on it. If you were to get this microbiology petri dish plate out of the package or make it yourself, a fresh made plate or a fresh pulled plate out of the package is going to look just like this. This is a sterile plate. There is absolutely no organism growing on this whatsoever, and this is what they look like. So your job is to put a bacteria species on it. You can see this has been done here as well as here. Over here you can see the species as well. And then incubate it for 18 to 24 hours and see what happens. Is it going to turn yellow? Is it going to turn pink? Is it nothing going to happen to it at all? Is it going to stay the exact same as it did to start with and nothing grow? All of that information is going to tell you what you're looking at, what type of species you've got going on, and how to better diagnose your species, or the species that you've got growing for your patient to treat your patient. Okay, so let's talk about how this works. So the very first thing that we need to talk about is that this agar is selective. So if this agar is selective, that means it's going to kill one particular species of bacteria or a group of bacteria, a large group of them. Now in this case, this particular agar, mannitol salt agar, is used specifically to test for the presence of gram-positive coccus bacteria. That's what this one is used for. So if you do a gram stain and you see a little round purple circle, that's the bacteria that you would put on this type of agar. And it's specifically used and designed to look for staph bacteria. That's what it's meant to look for. So the different species of staph are going to produce different colors to allow you to tell apart what species of staph they are. And this is how this works. So staph bacteria, they have they're not halophiles. They don't require lots of salt. They're osmotolerant. They can survive large swings in osmolarity, salt concentrations in their environment. It doesn't bother them so much. Most bacteria cannot do that. They'll dry out and die. So anyway, you put gram-positive coccuses on this plate, and they're all going to die. Pretty much anything other than Staphylococcus and a couple of other species will be killed instantly by the high salt level in this agar, 7.5% salt. If you cannot survive in that high salt content, you will be killed from the get-go. You're going to land on here, and you're going to touch all of that nice salty agar, and you're going to go, oh, this is really bad. All the water is going to instantly drive out of your cells into the agar. You're going to dehydrate. You're going to shrivel up into a little ball, and you're going to die. So this agar is designed to kill species of bacteria that cannot survive in this high presence of salt. So this salt, this 7.5% sodium chloride, is considered the selective agent. It is going to do the killing of certain species of bacteria. You can survive the salt, you're going to live. If the salt's too much for you, you're going to die. So the salt's going to be the selective agent, what is killing the bacteria in this agar. If you're not killed by the salt, you can grow. If the salt gets you, the salt has killed you. That's the only thing that would kill you is the salt. So that's how this works. This is the selective agent. Now, what type of species are going to be killed by the salt? Now, I mentioned this agar is used specifically for staphs. Pretty much everything other than staph is going to be killed by this salt content. Streptococcus species, they're gram-positive coccuses. They can't survive the salt concentration, so they will die. Bacillus species, gram-positive, will die. Most gram-positive species of coccuses will die other than Staphylococcus. So, almost all gram-negative species will die. Gram-negative bacteria, they cannot survive that high concentration of salt, like staph can. Staph goes on your skin, if you recall that. Your skin's very salty, and so they will die as well. So this is a gram-positive selective media. It is used for gram-positive coccuses. In specificity, it is used for Staphylococcus, which is a gram-positive coccus. So that's what this agar is used for. Now you know how to tell the staph, how it kills the staph from other species of bacteria. So now let's talk about the differential part. Well, if you can grow on this, we've automatically shown that you can survive that high presence of salt. So if you can grow, you're probably a staph species. Well, how do you tell apart the different species of staph that are growing on this agar? Well, you can see here we have one mannitol salt, one MSA plate, and these are both MSA plates as well. Part of it's yellow, part of it's pink, and part of it's still the same color that it was to start with. This whole plate's yellow over here, and this whole plate's pink. What's going on here? How did they go from this to this? Well, this is where that differential agent comes into place. In this case, the differential agent is the sugar mannitol. Can you eat the sugar mannitol or not? And that's what the differential agent is. The substance that's found inside of the media that bacteria will be able to eat or not. If you can eat it, you will produce a waste product, which will show up in the media. If you can't eat it, you'll eat something else, which will produce a different type of waste product, which will produce a different reaction. Okay, so let's talk about what does this. So mannitol. Mannitol is a type of sugar. Bacteria break down sugar through the process of fermentation. When bacteria ferment sugars, they always produce acids, some sort of acidic waste product. Vinegar is a perfect example of this. They eat the sugars inside of the apple juice and produce acetic acid. So bacteria eat the sugar mannitol. It goes through their metabolic process through fermentation, and an acidic waste product is produced. That acidic waste product will be released outside of the bacteria cells. It's waste, so they need to get rid of it. So they will release it outside of their cells, and it just kind of it's like putting a drop of acid on the plate. It just diffuses out into the agar. It just kind of they let it out of their cells, and it's just kind of going to go out into the agar and just diffuse out through the agar. So that acid, you can't tell apart acids from bases or water. They're all clear. Excuse me. So inside of this agar, there's going to be something that registers pH changes. And that's called a pH indicator. It's pretty much a litmus test, a little litmus strip. And in this case, the pH indicator is phenol red. Phenol red is this color under a neutral 7 pH. If you add acid to phenol red, it turns yellow. Anything other than 7, lower on the end towards the 0, is going to turn it yellow. The more yellow, the more acid, the lower the pH. So anyway, this bacteria, the Staphylococcus species, is able to eat the mannitol sugar. They ferment that mannitol sugar, produce an acidic waste byproduct. That acidic waste byproduct diffuses out through their cells, or is released through the cells, diffuses out into the agar, bonds to that phenol red pH indicator, and then turns it yellow. And that's how you tell that this particular species of staph is able to eat this agar, eat the mannitol. It's able to grow on this petri dish. We know that because it could survive, it grew in the first place. That means it can survive the presence of salt, which means it's probably some sort of staph. And we also know that because it's yellow, it was able to eat the sugar mannitol. It ate the sugar mannitol, fermented it, produced an acidic waste byproduct, which turned this agar with the phenol red indicator yellow. So this yellow color change indicates that this bacteria is able to produce an acidic waste byproduct through fermentation of the sugar mannitol. Now if you see here, this particular plate is pink. It started out the same as here, but this species of bacteria, this species of staph, is not able to eat the sugar mannitol. Now there are other foods in agars, and the default answer, as always, for every other species of agar, or every other type of agar pretty much on the planet, if they can't eat the differential agent, the mannitol, what else are they going to eat? The default answer is proteins. All the agars are pretty much rich in proteins. This one's one perfect example of that. This is also very rich in proteins. Bacteria, as well as pretty much every other organism on the planet, will go to proteins pretty much last to get their sources of energy. They're going to go for sugars first. So sugars are the low-hanging fruit. They're going to break down sugars. Well, if you can't eat the sugars, unfortunately, that's all you're left with are proteins. So if you can't eat the sugar, the bacteria is not going to starve. It's going to start eating the proteins in this medium. Now, this particular species over here, it could not eat the mannitol. It was struck on here, put in the incubator, couldn't eat the mannitol, so it started eating the proteins. Now, the process of breaking down proteins in bacteria is called hydrolysis, and hydrolysis of proteins produces a basic byproduct, a pH of 7 or higher byproduct. Now, a basic solution coming in contact with the indicator phenol red in this agar is going to produce a really, really, really dark pink color change. So you can see here that this particular species of bacteria was unable to eat the mannitol found in this agar. It did not starve. It started hydrolyzing the proteins found in this agar, which produced a basic waste product, which then turned the agar pink when it came in contact with that phenol red. So this pink reaction here, or this yellow reaction here, is what you're going to be looking for. Yellow reactions tend to be the bacteria Staphylococcus aureus. Not always. They tend to be this particular species of staph. This guy's pathogenic. It causes problems. The other one is Staphylococcus epidermidis, this pink color. It's what that usually tends to be. It's an opportunistic pathogen. It's going to be the one that causes you problems if you have a cut and it gets inside of you. Really nasty. Some people carry it. About 1 in 5 to 1 in 4 people naturally carry Staphylococcus aureus, but the ones of us that don't carry it, if you get it on you, it can cause some serious issues, staph infection-wise. This guy, pretty much everyone on the planet carries him. You should expect to find in there he's a commensalistic organism. So you take a swab from your skin, you swipe that swab on a mannitol salt agar plate or swab from your patient that has a staph infection, and you need to figure out which type of staph it is. You put it on there, you incubate it for 24 hours, you come back and you look to see what color change has occurred. Started out like this, you put some bacteria on it, and now you look at it and it's yellow. You look at that and you go, oh, it's yellow. This bacteria was able to eat the mannitol, meaning it produced an acidic waste byproduct. That is probably Staphylococcus aureus. this particular patient has a pathogenic form of a staph infection. I need to go with this type of treatment. Or you can look at it and get the pink and go, they have a commensalistic staph infection. Need to go with a different type of treatment. So this is the little quick rundown of how mannitol salt agar works. Here's a YouTube video once again of someone going over that as well. Now, one quick little caveat for medias. Medias have a shelf life in the incubator. They only, like I said, can last for 18 to 24 hours in the incubator. And the reason for that is they run out of sugar. This particular bacteria is Staphylococcus aureus. This is a pathogenic form of staph. If you did get this yellow, you're good to go. Now, if you come back about 30 hours later, you forgot about your plate, you left it in the incubator, this Staphylococcus aureus has eaten all of the mannitol inside of that petri dish, produced a beautiful yellow color. It ran out of mannitol. At about 25 hours, it ran out of mannitol inside of that petri dish. There's only a certain amount of mannitol inside of there. It ran out of mannitol. And it's not going to starve to death. It's going to switch over to eating the default answer, the proteins. So that Staphylococcus aureus, which eats the mannitol, will then start hydrolyzing that protein, producing a nice basic waste product, which will neutralize all of the acidic waste product that it produced earlier, turning the plate eventually pink. So what happens is it can mess up your diagnosis. So anything after 18 hours, 18 is really the best, 24 is kind of the most. Anything after 24, you run the risk of cross-diagnostic, the plate not being reliable anymore because the bacteria ran out of sugars and ran out of substances to eat. So this is mannitol salt agar, a selective media that is used for gram-positive coccuses. It is selective against gram-negative bacteria, which means that's what it kills. Its differential agent is mannitol. Uses the concept of acid versus base with the pH indicator phenol red. For your lab exam, I would expect you guys to know what the selective agent is, what the differential agents are, in these types of agars. I would also expect you to be able, if I posted a picture of a yellow plate or a pink plate, to go, oh, this bacteria is able to ferment the sugar mannitol, or this bacteria is able to hydrolyze proteins. And I know that because blank, and you could fill in the answer to that. I know that because it produced a yellow color change, which means that it produced an acidic waste byproduct for the fermentation of mannitol sort of thing.